हेलो एवरीवन, वेलकम टू माय चैनल। जैसा कि आप सभी ने इंट्रो वीडियो में देखा होगा कि मैं अपने चैनल पे क्या क्या करने वाली हूँ, तो मुझे यहाँ बताने की ज़रूरत नहीं पड़ेगी। मैं बिना वक्त ज़ाया किए इस चैप्टर को स्टार्ट करती हूँ, जो आज का हमारा चैप्टर है, दैट इज़ बायोलॉजिकल क्लासिफिकेशन। Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that didn't. In Linnaeus' time, a two kingdom system of classification with Plantae and Animalia kingdom was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic, that is green alga, and non-photosynthetic, that is fungi organisms. Classification of organisms into plant and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of cell, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification system for the living organisms have hence undergone several changes over the time. Though plants and animals kingdoms have been a constant under all different systems, the understanding of what group slash organism be included under these kingdoms have been changing. The number and the nature of the other kingdoms have also been understood differently by different scientists over the time. Characteristics of five kingdoms. First one is cell type. In Monera, that is prokaryotic. In Protista, that is eukaryotic. In fungi, that is eukaryotic. In Plantae, that is eukaryotic. And in Animalia, that is eukaryotic. Next one is cell type. In Monera, non-cellulosic, that is polysaccharides plus amino acid. Next one is Protista, present in some, present with chitin in fungi, present with cellulose in Plantae, and absent in Animalia. Next one is nuclear membrane. In Monera, it is absent, and in Protista, fungi, Plantae, and Animalia, it is present. Next one is body organization. In Monera, it is of cellular type. In Protista, it is cellular type. In fungi, it is a multicellular slash loose tissue. In Plantae, it is a tissue slash organ type. And in Animalia, it is a tissue slash organ system. The next one is mode of reproduction. In Monera, mode of reproduction is autotrophic, chemosynthetic, and photosynthetic, and heterotrophic, saprophytic, and parasynthetic. In Protista, autotrophic, photosynthetic, and heterotrophic. In fungi, it is heterotrophic and saprophytic and parasitic. In Plantae, it is autotrophic, photosynthetic, and in Animalia, it is heterotrophic, holozoic, and saprophytic, etc. R.S. Whittaker in 1969 proposed a five-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for the classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 gives a comparative account of different characteristics of five kingdoms. The tree domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain and thereby a sixth kingdom classification. You will learn about this system in detail at higher classes. Let us look at this five kingdom classification to understand the issues and consideration that influence the Classification system. Earlier classification system included bacteria, blue green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae, that is cyanobacteria, with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Say for example, Chlamydomonas and Spirogyra was placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants. So they all show a characteristics difference in their walls composition, the fungi and the chitin in their cell wall, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in a kingdom Protista. Kingdom Protista has brought together Chlamydomonas and Chlorella, earlier placed in algae within plants and both having cell walls, with Paramecium and Amoeba that which were earlier placed in the animal kingdom which lacks cell wall. It has put together organisms which in earlier classification were placed in a different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future to depending on the improvement in our understanding of characteristics and evolutionary relationship. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities but is also phylogenetic, that is based on the el- evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whittaker system of classification. The kingdoms Plantae and Animalia, commonly referred to as the plants and animals kingdoms, respectively, will be dealt separately in chapter 3rd and 4th. Kingdom Monera. Members of kingdom Monera, they are most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot spring, deserts, snow, and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as a parasite. Bacteria are grouped under four categories based on their shapes: the spherical coccus, that is cocci; the rod-shaped bacillus, bacilli; the comma-shaped vibrium, that is vibrio; and the spiral spirillum, that is spirilia. Figure 2.1. Bacteria of different shapes. First one is cocci, second one is bacilli, and in bacilli there is a spore. Third one is spirilia, and in spirilia there is a flagellum, and fourth one is vibrio. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrate. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on another organisms or on dead organic matter for food. So next one is archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, that is halophils, hot springs, that is thermoacidophils, and marshy area, that is methanogens. Archaebacteria differ from the other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cow, buffaloes, and they are responsible for the production of methane, that is biogas, from the dung of these animals. Next one is eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, that is also referred to as the blue-green algae, have a chlorophyll A similar to green plants and are photosynthetic autotroph. That is figure 2.2. The cyanobacteria are unicellular, colonical, or filamentous, and freshwater and marine water or terrestrial algae. The colonies are generally surrounded by a gelatinous sheet. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocytes, that is nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substrates such as nitrates, nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume. And there is a diagram given, that is a filamentous blue-green algae, that is nostoc. In roots, etc., some are pathogens causing damage to human beings, crops, farms, animals, and pets. Cholera, typhoid, tetanus, citrus, cancer are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission, that is figure 2.3. A dividing bacteria is diagram is given. Sometimes under unfavorable conditions they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to another. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Next kingdom is kingdom Protista. A single cell eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protestant to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the others dealing with plants, animals, and fungi. Being eukaryotic, the protestant cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protista reproduce asexually and sexually by processes involving cell fusion and zygote formation. Algae, that is bestmates, they are found in fresh water as well as in marine environments. They are microscopic and float passively in water current, that is called planktons. Many of them are photosynthetic in diatoms. The cell wall forms two thin overlapping shells which fit together as in a soap box. The walls are embedded with cilia and thus the walls are indestructible. Thus diatoms have left behind large amount of cell wall deposits in their habitats. This accumulation over billions of years is referred as a diatomaceous earth. Being gritted, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producers in the oceans. Next one is dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, red, depending on the main pigments present in their shells. The shell wall has a stiff cellulose plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between wall plates. Very often, red dinoflagellates, example Gynoclics, undergo such rapid multiplication that they make the sea appear red, that is, red tides. Toxin released by such large number may even kill other marine animals such as fishes. Next one is euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicule, which makes their body flexible. They have two flagella, a short and a long one. So they are photosynthetic in presence of sunlight. When deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants, that is example euglena. Next one is slime molds. Slime molds are saprophytic protists. The body moves also along decaying twigs and leave engulfing organic material under suitable conditions. They form a aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies bearing spores at their tips. The spore passes through walls. They are extremely resistant and survive for many years. Even under adverse conditions, the spores are disappeared by air currents. Next one is protozoans. All protozoans are heterotrophs and lives as a predator or a parasite. They are believed to be primitive relatives of animals. There are four major groups of protozoans. First one is amoeboid protozoans. These organisms live in a fresh water, seawater, or a moist soil. They move and capture their prey by putting out pseudopodia, that is false feet as an amoeba. Marine forms have cilia cells on their surface. Some of them, such as entamoeba, are parasites. Next one is flagellated protozoans. The members of this group are either free living or parasite. They have flagella. The parasitic forms cause disease such as sleeping sickness. Example, trypanosoma. Next one is ciliated protozoans. They are aquatic, actively moving organisms. Because of the presence of thousands of cilia, they have a cavity that is called a gullet that opens to the outside of the cell surface. The coordinated movement of a row of cilia causes the water laden with food to be steered into the gullet. Example, paramecium. Sporozoans. This include diverse fungi. Diverse organisms that have an infectious spore-like stage in their shell wall. The most nutritious is Plasmodium, that is malarial parasite which cause malaria, a disease which has a staggering effect on human population. Kingdom is kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on the moist bread and rotten fruits. The common mushroom you eat and toadstools are also fungi. White spots seen on mustard leaf are due to a parasitic fungus. Some unicellular fungi, that is yeast, are used to make bread and beer. Other fungi cause the disease in plants and animals. Wheat rust causing Puccinia is an important example. Some of the source of antibiotic, that is penicillium. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infections. With the exception of yeast, which are unicellular, fungi are filamentous. Their bodies consist of long cylinder thread-like structures called hyphae. The network of hyphae is known as a mycelium. Some hyphae are continuous tube filled with multinucleated cytoplasm. These are called cenocytic hyphae. Others have septate or cross walls in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrate and hence are called saprophytes. Those that depend on living plants and animals are called parasites, though they can live as the symbiotics in association with algae as a lichen and with roots of higher plants as a mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spores called conidia or sporangiospore or zoospore, and asexual reproduction is by oospores, ascospore, and basidiospores. The various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following three steps. First one is fusion of protoplasm between two motile or non-motile gametes called plasmogamy. Second step is fusion of two nuclei called karyogamy. And third step is meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae of a compatible mating types to come together and fuse. In some fungi, the fusion of two haploid cells immediately results in a diploid cell that is two men. However, in other fungi, that is Ascomycetes and Basidiomycetes, an intervening dikaryotic stage that is n plus n, that is two nuclei per cell, occurs. Such condition is called as a dikaryon, and this phase called dikaryophase of fungus. Later, the parental nuclei fuse and the cell become diploid. The fungi form protein bodies in which reduction division occurs, leading to formation of haploid spores. The morphology of mycelium, mode of spore formation, and fruiting bodies form the basis for the division of the kingdom into various classes. First one is Phycomycetes. Members of Phycomycetes are found in aquatic habitat and on decaying wood in moist and damp places or as a obligate parasite on plants. The mycelium is aseptate and cenocytic. Asexual reproduction takes place by zoospore, that is motile, and by aplanospore, that is non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphological, that is isogamous, or dissimilar, that is anisogamous or oogamous. Some common examples are mucor, figure 2.5a, rhizopus, that is the braid molded mentioned earlier, and that is albugo, the parasitic fungi on mustard. Next one is Ascomycetes, commonly known as the sac fungi. The Ascomycetes are mostly multicellular, that is penicillium, or rarely unicellular, that is yeast, that is Saccharomyces. They are saprophytic decomposers, parasitic or Coifophilous, growing on dung. Mycelium is branched and septate. The asexual spores are conidia produced exogenously on the special mycelium called conidiospores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac like asci, that is in singular it is ascus. These asci are arranged in different types of fruiting bodies called ascospores. Some examples are aspergillus, clavipsipus, and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered as a delicacy. Next one is Basidomycetes. Commonly known as Basidomycetes are mushrooms, fungi or puffballs. They grow in soil, on logs and tree stumps, and in living plant bodies as a parasitic, that is rust and smuts. The mycelium is branched and septate. The asexual spore are generally not found, but vegetative reproduction is by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately give rise to basidium, that is karyogamy, and meiosis takes place in the basidium, producing four basidiospores. The basidiospores are exogenously produced on the basidium, plural basidia. The basidia are arranged in a fruiting bodies called basidiocarpus. Some common members are agricus, that is mushroom, astilgo, that is smut, and buxine, that is dust fungi. Last one is Deuteromycetes, commonly known as the imperfect fungi because only the sexual or vegetative phase of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they are rightly belong to. It is also possible that the sexual and vegetative stage have given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out to the Deuteromycetes. One perfect sexual stages of the member Deuteromycetes were discovered. They were often moved as Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of a litter and help in a mineral cycling. Some examples are Alternaria, Coletrichicum, and Trichoderma. All eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic, such as insectivorous plants or parasites. Bellderwort and Venus flytrap are examples of insectivorous plants, and Coscuta is a parasite. The plant cell have an eukaryotic structure with prominent chloroplast and cell wall mainly made up of cellulose. You will study the eukaryotic cell structure in details in chapter 8. Plantae includes algae, bryophyte, pteridophyte, gymnosperm, and angiosperm. Life cycle of plants has two distinct phases: the diploid saprophytic and the haploid gametophytic. They alternate with each other. The lengths of the haploid and diploid phases and whether these phases are free living or dependent on other vary among different groups in plants. This phenomena is called alternation of generation. You will study the further details in of this kingdom in chapter 3. Next one is kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack cell wall. They directly or indirectly depend on a plants for food. They digest their food in an internal cavity and store food reserves as a glycogen or a fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms so elaborate sensory and neuromotor mechanism. Many of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Silent features of various phyla are described in chapter 4. Effects a virus can have on us. Even if we do not associate with our condition, virus did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. That is the diagram of tobacco mosaic virus, that is TMV, and bacteriophage. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? Virus mean vermin or poisonous fluid. Dmitry Levonsky in 1892 recognized certain microbes as a causal organism of the mosaic disease of tobacco, that is given in figure 2.6a. These were found to be smaller than bacteria because they pass through bacteria-proof filters. M.W. Birkin in 1898 demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants, and named the new pathogen virus and called the fluid as a contaminum vivum fluidium, infectious living fluid. W. M. Stanley in 1935 showed that the viruses could be crystalline, and crystals consist largely of proteins. They are inert outside their specific host cells. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No virus contains both DNA and RNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single stranded RNA, and viruses that infect animals have either single or double stranded RNA or double stranded RNA. Bacterial virus or bacteriophages, virus that infect the bacteria, are usually double-stranded DNA virus, that is given in figure 2.6b. The protein coat called caspid, made up of small subunits called caspospores, protects the nucleic acid. These caspospores are arranged in a helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptoms can be mosaic formation, leaving, rolling and curling, yellowing and vein clearing, dwarfing, and stunt growth. Viroids. In 1971, Theo Diner discovered a new infectious agent that was smaller than virus and caused potato spindle tuber. It was found to be a free RNA. It lacked the protein code that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by agent consisting of abnormally folded protein. The agent was similar in sizes to virus. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy (BSE), commonly called mad cow disease in cattle, and its analogous called variant Cia tobacco disease, that is CJD, in humans. Lichens. Lichens are symbiotic association, that is mutually useful association between algae and fungi. The algal component is known as phycobiont, and the fungal component is as a mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi, and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is the association, and if one so is lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicator. They do not grow in pollutant area. That's all. Thank you for watching this video. Bye.